recording. Cool. Okay, so yesterday, beginning of class, or Tuesday to end class, what I should have tossed in here is some language that I hadn't shown you before, but concepts we have kind of talked about. So when we were making the Punnett squares for the freckles coming off the Weasleys, we had talked about, we looked at this and said, okay, well, we know what mom is, we know what dad is, and we can make a comment about what all the kids are, and we said that in this case, all the kids would have freckles. Another way to say that word all is to use a percentage. So we could have said 100% of the kids will have freckles. The very literal definition of the word all. Down here, when we were talking about Punnett squares, we said, okay, well, we know what mom is heterozygous, dad's heterozygous, so we filled in the Punnett square, and we said, okay, well, this, and this are all going to result in freckles. This one is not going to result in freckles. And so we put together that 3/4 of the kids, statistically, would have freckles and 1/4 would not. And we had fractions and we had percentages. There is language in math to talk about fractions or percentages without using the word fraction or percent. What's the other word in math that we use to talk about fractions and percentages? A ratio. So these are ratios. Ratios of what? Okay, never mind. So these are ratios of whether or not someone has freckles or someone doesn't have freckles. The having freckles or the not having freckles, these are observable traits, yeah? What do we call observable traits when we're talking about genetics? Yeah, but what do we call them? They have a name. A P, but it sounds like an F. And so what we've done here, without actually calling it this, by saying that 3/4 or 75% of the kids are going to have kids and 1/4 or 25% of the kids are not going to have freckles, what we have created is what's called a phenotypic ratio. It's a ratio of, or a set of ratios of potential phenotypes expressed in the offspring. And so this is one of the concepts that you would have needed for 2 or 3 of the questions in the homework that I gave you. This language that when you separate out phenotypes and give them percentages or probabilities, what you've created are phenotypic ratios. Yes, Brooklyn. Yes. Your ratios need to add up to 100. Yes. So that's a phenotypic ratio, but instead of looking at the phenotypes, we could have also looked at what the genes actually are when they're expressed. So we have 3 options here. We've got big F, we've got the heterozygous option, and then we've got the little f option. How many of the offspring are going to be homozygous dominant? 1 out of a possible 4. How about the little f? 2 out of 4, which we can reduce to, sorry, big F, little f. The big F, how many big F, little f kids could I have here? 2 out of 4, which we could reduce to, that's a reducible fraction, to a half. Half of the kids could be heterozygous, which would leave us with 1/4 being homozygous recessive. Looking at the phenotypes, we create phenotypic ratios. Looking at the genotypes, we create genotypic ratios. Genotypic ratios. And you actually did this without knowing that language. You did it, if you did the homework, you did it a bunch of times right near the end. Everywhere where it asked you to say, okay, well, what percentage is this, and what percentage is that, and what percentage does it look like this, and what percentage looks like that. I know this because I just did them. You were, without knowing it, you were creating genotypic and phenotypic ratios. So let's go through the homework I gave you just to make sure we're feeling okay about this stuff. This is what we need here. Okay, so the practice problems started here. This is what I got for this page. A, B, and C in each of these questions just kind of set up what it's asking you to do. And then D and then you fill in the Punnett square. And then D and E actually get you to talk about offspring. Compare what you got with what I got. Comments, questions, concerns on this page. That is hot coffee. At the very least, you and I are in agreement. Again, don't assume that if you got something different than I got, that you're wrong. As Kaylee has demonstrated over and over and over again, I make mistakes. I'm not resentful about that at all. I like that someone finds my mistakes. One of my physics 11s waited till I got to the very, very end of a long problem to point out that I'd made a mistake in step 2. Don't do that. If I make a mistake, point it out quickly, please. That would be great. Okay, we're good? Okay. Here's this page. I'll give you the top 3, and we'll take a look at the bottom one. Again, oh, sorry, Evelyn. What are you looking at? The first one I'm talking on. In the flower position, that says stem, because the options are either stem or tip. Yeah? Yeah, they're either along the stem or at the tip. No, because along the stem is the dominant trait, so both parents would express only along the stem. Because both parents are heterozygous. Oh, the children part. 3/4 would be on the stem and 1/4 would be on the tip. I didn't include the words along or at. I just gave the positions. As long as you can understand it, and then are able to write it less confusingly on a quiz or a test, that'd be great. Before I forget, Asia, I should mark you here. Boom, there you are. Okay, here's the bottom one. Rabbit fur. Black is dominant, white's recessive. Actually, unfortunately, it gets more complicated than that. I'm going to show you guys something today that I'm not going to require you know, but I want to show you how complicated this stuff actually gets. Okay. Comments, questions, concerns? Once, twice, thrice, sold. Okay, cool. Here we go. So I told you yesterday, a bunch of you asked, you were not able to do questions D and E, but you can now. The comparison of the number of each phenotype is, if you're comparing numbers of phenotypes, what is that? The phenotypic ratio would be the next one, yeah. This one should be genotypic. Ratio. Okay, so here is the language for the problems that are on this page. Probability F1, F2, phenotypic, genotypic, homozygous, homozygous dominant, homozygous recessive, heterozygous. And then the purpose of the Punnett square is to determine the probabilities of the children's geno and phenotypes. Couldn't hurt. I mean, presumably one will give you the other. If you know the genotypes, you can deduce the phenotypes pretty easy. Yes. Just so long as you understand that there's technically more. Okay. Questions, comments, concerns? I mean, we could also use them to figure out what parents are. If you said, did anyone say anything differently in there that you want to discuss? Again, just because we got something different, don't assume that you're wrong. Okay. Here is this page. So this whole page comes down to this teeny tiny little Punnett square here in the middle. You've got two heterozygous P parents. And they're going to make a offspring, presumably lots because they're plants. I labeled all of these different parts using the language that was in this box here. Comments, questions, concerns about anything I did there. Going once. How do I get what? Because when I introduced Punnett squares right at the beginning, I said 4 monohybrid crosses. This is a tool that is what that means. A monohybrid is a single trait being crossed. I'm going to show you something today that is the next logical step in this, just to show you how crazy some of this stuff gets. We are not going to do it. Like, I'm not going to require you to follow and to, learn it and then to regurgitate it. Like, I'm not going to expect that of you, but I want to show you what this stuff, where it goes next. Yes, Brooklyn. Okay, so we're moving down the page. Okay. That would be fine as well. Yes, that would be totally acceptable as well. Big R, little r, big R, little r. Yeah, you bet. Yeah, that would be totally acceptable. The phenotypes. So then the other ones I didn't answer were down there at the bottom, E and F, which we can answer now. What's the genotypic ratio of the F1 generation and the phenotypic ratio of the F1 generation? So the genotypic ratio is going to be 1/4 big R, big R. It's going to be 1/2 big R, little r. And it's going to be 1/4 little r, little r. Or you could give them as percentages, 25, 50, 25. Whether it's a fraction or percent, I don't care. Or a decimal if you're crazy. And then the phenotypic ratios we already have. What percentage of the offspring will have round seeds? What percentage of the offspring will have wrinkled seeds? This is this. 25, well, the whole thing. It's 75% wrinkled, or round rather. 25% wrinkled. That's the, putting both of those things together in one expression is the phenotypic ratio. And then if I'm not mistaken, on the next page, you guys actually would have gone and done all of this without actually talking about genotypic and phenotypic ratios. That's what this is. What percentage were all the genotypes? What percentages were the various phenotypes? So you did all that without actually knowing the language, assuming you got this done. So let's let this sit here. Take a look at what you've got, compare it with what I've got, assuming you went through and did it. Are there any comments, questions, concerns? Which one? Sorry. Oh, don't. Oh, that's what you, for number one, that's how you answer number one? What was your answer for number one? So the only thing I would add to that is when both. The only way for a recessive trait to be expressed is if both alleles are lowercase. We'll get there. Are we cool with the first three? Evelyn. Oh. You need at least one uppercase. So that's either an uppercase, lowercase, or it's two uppercases. By definition of dominant, you only need one to pound the other one into the ground. Like my daughter and my son. Yet another example yesterday of when she drove his face into the ground. It's good times. And then he screams. It's good times. Okay, Evelyn, you want to talk about the bottom ones? Let's do it. So these should all be big T. So what do you want to talk about? Okay. Oh, you were out of the class yesterday when I explained that. Yeah, no, definitely. Yeah, you were. It's when you and Brooklyn weren't here. I did explain it in the video, so if it was confusing, you can always go back and look at the video. Ooh, maybe that wasn't in the video. I can't remember if I hit pause or not at that point. I don't remember. Sorry? Yeah, I'll make that clearer. Should be 25, 50, 25, and then 75, 25. Okay, next page, we good? Jedediah's falling asleep, so we're going to speed up. Okay, here's this one. I didn't fill out this punted square because it's as easy as the first one was. They're all lowercase, so it's going to look like that. Can I slide this up and do it all? Yes, I can. That's the whole page. Comments, questions, concerns about anything on this page? Can you be more specific? No, it doesn't. Like, it doesn't, like, where do you put the parents? As long as one's on the top, one's on the side, it doesn't matter. I usually go dad, mom. That's just my habit. I typically am pretty consistent that way. Doesn't matter. Okay, and then I heard comments at the beginning about the hairy toad trolls. Let's talk about some trolls. So this is where I got to yesterday. Sorry, this morning, as you guys were coming in. I didn't get a chance to finish this, so let's talk about it. Imagine three pairs of trolls and their offspring are discovered on a remote island. Phenotypes of the parents and their offspring are shown in the table below. So you can see that in the first pair, both parents have hairy toes. In the second pair, the male has hairless toes and the female has hairy toes. And in the third, they both have hairy toes again. And then their offspring of the first pair are 75-25, hairy, hairless. The hairless hairy gave us 50-50, which is interesting. And then this hairy pair gave 100% hairy toes. At this point, I think I've said the word hairy enough so that it's lost all meaning. I figured, and I hope that you're able to follow through too, that hairy seems to be the dominant trait here. Because if you have a hairy toad and a hairy toad parent and a hairy toad parent, and to get 100% hairy toes, you need to recognize that there's two ways for that to happen. Nope. Nope, that's not the one. You know what? Let's just start making things and see what happens. There has to be a lowercase somewhere. So if we're right, the easy one to do is to do pair number 2. This is the easy one. If we are correct that the hairless toes are the recessive gene, then we can say, okay, well, let's go little h for the male. We'll go I'm not sure why it's given us, yeah, interesting. Because we don't know what this female could be. I think we need 2 for pair 2. Because this one right here and this one right here, this one especially, seems to suggest that for there to be a hairy toad and a hairy toad, for 75% to come out hairy toad and 25 to come out hairless toad, what you'd have to have there is a scenario where you have this, where you have big H, we're going to use H for hairy, big H, little h, and then little h, little h. This is the kind of scenario where you have 75% hairy toad, 25% hairless toes. And if it was the other way, that the hairless was dominant, then this was not what the table would look like. Because if the hairlessness was dominant, then these two would go the other way. The heterozygous ones would flip. So then for pair number 1, we can see here that we don't know necessarily which parent, that we cannot determine. But one parent has to be full-on dominant for there to be both hairy and yet have this percentage breakdown. And one parent has to be heterozygous. Sorry, nope, you're right. That makes no sense. It doesn't make any sense at all. You're right. They both have to be heterozygous. Yes, you're right. Thank you. They both have to be heterozygous to get this bottom one. Yes. Of the offspring, you mean? 1 and 4, yeah. I get it. Or per. That child. The next one, the pair 2, the hairless hairy. This is the one that could go, well, for it to be 50-50, and if all of them have a little h in there somewhere, to have 50% of these be hairless means we need to have 2 little h's somewhere. And for the other half to be hairy, we have to have 2 big h's somewhere. So the mom in pair 2 has to be what? Yeah, mom's got to be heterozygous. Dad's got to be full-on recessive, and mom's got to be heterozygous. Or other way around, right? Doesn't matter which one, male or female. That's the only way we're going to get a 50-50 phenotype split. And then in pair 3, 100% of them have hairy toes. So the easiest way for 100% of them to have hairy toes is if this is true, is if both parents are dominant, because then you're going to get H's everywhere. But the other way for it to happen is if you get one big H everywhere, but we know actually that both parents have hairy toes. So one has to be full-on dominant, the other could be full-on dominant, or the other one could be heterozygous. Because the heterozygous would give big H, and then big H, little h, big H, little h. Brooklyn, does this clear up some of your concerns? Okay, cool. And so then we can use this to figure, well, we know the genotypes of parent 1, they both have to be heterozygous, or both big H, little h. Pair 2 has to be big H, little h, and little h, little h. And then 3 could be either big H, or big H, little h. And that is where your homework ended. Basically, we just use those ratios to go backwards. Questions, comments, concerns? Any clarification needed? Going once, twice, thrice. Sold. Okay, cool. Put your pencils down or pens down, your writing utensils down for a second. I am going to show you not first what we're going to do today, but where the crossing can get to. I at least want to expose you to this. So, those were Punnett squares for monohybrid crosses, and that's largely what we're going to do. But it doesn't stop with monohybrid crosses. This is what your friends in pre-div science 10 are going to take a look at. You guys get on the pass on this one. So, mono means one. What if there's more than one trait that you're crossing? Well, those are called dihybrid crosses. And so we can start looking at, okay, well, I've got multiple traits being expressed in these animals. What if I wanted to start breeding a multiple traded cat with a multiple traded kid? It's not just black and white anymore. What if it's like brown with a short tail, white with a short tail, the short tail being dominant, white being recessive, and then brown being dominant with a long-tailed cat, long-tail being the recessive gene. I'm breeding these two things together. Now all of a sudden we're comparing two sets of traits, and the genetics for this gets a little bit more interesting. You have to create what are called dihybrid Punnett squares, and instead of being 2 by 2, I'm going to show you that those would have to be 4 by 4, which gives you 16 possible offspring. The number of traits that we're crossing. Okay, we're getting closer to the real one, yes. Closer. So, it all comes down to gene placement on chromosomes. If two genes are on different chromosomes, all 4 possible allele combinations for 2 different genes, there's a blanks here, because this is actually the PowerPoint slide bank that Mr. Easton used to use for his pre-diploma. He gave it to me. He's like, I just want to see what you used to do. I want to show it to my students. Yeah, go for it. So I don't have all of the blanks, but that's okay. I'm going to try and explain to you as much as I can. So if they're on different traits, or if they're on different chromosomes, then when the copying happens and these things line up inside like the process of meiosis, this is why you learn meiosis in 9, so we can talk about stuff like this in 10. When they line up, they can line up on either side as they're getting split. And so you get multiple possible combinations of gametes being produced in the males and the females of these different cat species, or whatever they happen to be. No. The only difference between a male and female at this stage is when meiosis has occurred. In males, meiosis is constantly happening. In females, meiosis only happens as an embryo. When you're born with all the eggs you'll ever have. That's the only difference here. If you are a biological female and you got 2 eggs, you got 2 chances to get pregnant and you're done. Which would be terrifying because that means either your first period or 28 days later is it for you, and then you're going into menopause at like 14 years old. That's the other scary part is what? It's basically yeah. As soon as the eggs are gone, there's no periods. I mean, that's the purpose of a period, right? To create a home for an egg. You run out of eggs, you're done. That's the purpose of menopause. Yeah. So, yeah. There's a ton of different reasons for it. Depends on the reason. So like, some people are infertile because there's no place, there's so much scar tissue on the inside. It's called endometriosis. They still get their periods. The periods are wildly painful. But they're infertile because the egg has nowhere to go because their uterus is covered with scar tissue. I had a student who I taught her every year 9 to 12. And once a month for a little bit, she would, a couple days, every month, she'd miss class because she'd have these ridiculous periods.
then that's, again, you learn pretty early on. If you don't know what endometriosis is, you don't have it probably. The other thing is, with that little fun fact, here's a side effect of birth control they don't necessarily tell you. Different birth controls can affect your body different, especially the pills, specifically different medications can affect your body differently. And so what some pills do is that they actually shorten that window, your period window, so you still get periods, but it's way shorter. The egg just like, shoo, see you later. And there's nothing like, the window is so small of you getting pregnant, it's not gonna happen. Another way for it to happen is it just stops your body from having periods. So those eggs get stacked up. So your body's just like, it gives you the hormone, and your body's like, it tricks your body into thinking it's pregnant. When you're pregnant, your body doesn't release eggs. So it's just like, let's just block off the dam, hold on to those eggs because we're pregnant, but you're not. And then there's women who are on this pill for years, which could be problematic. One of the side effects of the speed up the release pill, again, I don't know what the names of these things are. I just know they mess with your hormone. There's one, again, the one that causes the period to happen rather quickly, can actually trigger your body to release more than one egg at a time. So, and there is no upper limit to how many you could theoretically force your body to release. So if you're particular, again, I don't know what kind, if you have a conversation with your doctor, it is possible, there's a possible side effect of a particular form of birth control, is that your body could theoretically release huge amounts of eggs all at once. No, we're talking like hundreds. But the pill is preventing you from getting pregnant. So you just lost, because you only have a limited amount of eggs in your body, hundreds of possible pregnancies in the future. And if your body releases too many in a shot, and does that a couple times, now all of a sudden, you were trying to delay pregnancy. Maybe you're like, I don't want kids at 60. Good choice. So I'm on birth control at 60. Excellent choice. But a possible side effect is the fact that at 18, the side effect kicked in, and now by 21, you have no eggs left. And now you're in your 20s going through menopause. And you will never have children. So there are these side effects do exist. Brooklyn.
When you said that like multiple eggs is that how like twins?
That's one way for twins to happen, yes.
So like if like
And your bliss is possible. Did you hear about the crazy lady who had like 7 or 9 at once? Yeah. So she was taking like intense fertility meds that caused her body to release a ton of eggs at a given shot. And there is a lot of sperm that were present at the time. And so every egg, we're like, hey, you! And the egg, okay. And so every egg got fertilized. She got pregnant with like 7 or 9 or whatever the ridiculous. Yes, all at the same time. Wildly. First of all, keyword, not stomach. Definitely uterus.
Yeah, like she like.
Definitely not your stomach. Abdomen. Your abdomen. Yeah.
Did she get early?
Oh, she had to, yeah. Yeah. They get delivered as early as humanly possible. So like 20 weeks. 20 weeks is the earliest possible. Well, again, there are outliers, but 20 weeks it's generally safe. Terrifying, but the odds of survival after 20 weeks are way bigger than ever.
So I have a question. So does that mean longer the period, more chances you like a little more? Like,
not necessarily. Not necessarily. To answer some of these questions, if you have questions about your own cycles, no, I'm not going to answer them. Please don't ask me. What I was going to say is, there are apps that I discovered when I got married, because my wife has them on her phone, that do a really good job of predicting with reasonable accuracy a lot of the details that you don't see behind the scenes. No, you don't. Not the couple apps my wife has. No, but things like, anyway, we'll leave that comment to simmer and percolate. We're going to come back to this. as a married dude with 2 kids, I learned a lot of things I didn't know I needed to know. Okay, so genes on different chromosomes. Meiosis is an important detail. You learn about it at 9, like why the heck does this matter? It matters. Now we're breeding things. So you're not going to discuss with your partner, but if you're setting up dihybrid crosses, there's actually a whole mess of ways your Punnett squares can show up. And again, they're all now going to be 4x4, not 2x2. So you're going to start getting situations that look like this. You can have a big R, big Y, big R, little R, big Y. Like these are all possible gametes from one parent. These are all the possible gametes from 2 parents, and these are just for the heterozygotes. And these are all of the possible offspring combinations you could have. So now you're starting to get really interesting genotypic ratios and phenotypic ratios. You've got 4, 5, 6, 7, 8 of them are all going to be round in yellow. 3 of them, oh wait, did I miscount that? 4, 5, 6, 7, 8, 9. 9 of them are going to be round in yellow. 3 of them are going to be round in green. 3 of them are going to be yellow and wrinkly, and one of the 16, 1 in 16 is going to be wrinkly and green. So things get really quite interesting here. Where things get really stupid is there's exceptions to all these laws. And Mendel did this stuff too. This was part of what Mendel looked at. So we talked about Mendel and his monohybrid crosses. He also did these dihybrid crosses. I told them. I have no. There's no reason why it couldn't get bigger. So, there is something that can happen during meiosis called recombination. Did anyone remember their Science 9 teacher talking about recombination? If you had me, it didn't happen. O mite, Easton mite, Morrison mite. Broad probably did not. But I know those 3 guys sometimes do. So, one of the fun little facts during meiosis is that things don't just copy the way we expect them to copy. Because all your chromosomes have a little centromere in the middle. And at the centromere, and in other places of the chromosome, you can actually have chromosomes like, so this is the homologous pair. Each half of that homologous pair gets copied. But then you can have scenarios where you have 2 of the white copy and 2 of the blue copy, and they just get close enough together and intertangled, and then the backbones break and they get reattached on the wrong pieces. So you get a piece of the white that has the blue and a piece of the blue that has the white, just for fun. That can happen. Nope. We're talking about individual chromosomes here. And so these can be the gametes. Where you would have expected it to be white, white, blue, blue, you can have white and blue, but then you can have these 2 just kind of split and traded for reasons I couldn't possibly fathom. Right? That can happen. And when that happens, you start to get some of these crossing effects that start to get weird. Things like, I wasn't expecting this possible gap or this possible genotype or phenotype to show up, but it can happen. So, you can start to get crosses you did not expect popping out on something called what are called linked genes. And then we're going to talk about it near the end of this chapter. Genetics and blank. Blank, and you should know the answer to that blank. Blank can change the allele frequency. So, for example, black is a dominant rabbit fur color. White is the recessive. But if you live in the Arctic, is it beneficial for you to have black fur? No, when the ground's white and you're black, you stick out like a sore thumb. And everything wants to eat you. The bears, the foxes, birds, everything. You are going to get consumed. And so as you get eaten, even though black was the dominant, what's going to happen over time is that the black ones are all going to die out. And the white one, the recessive trait, is all that's left. What's this called? When nature selects a particular trait. No, it's not called Mother Nature. I said the words. When nature selects a trait.
Environmental breeding.
Amatos, you smiled, so you know the answer. Yeah, it is. right at the bottom. Natural selection. Natural selection is a concept that, even if you don't know a lot about it, you should be familiar with the idea that there are traits that nature will just select for beyond what genetics says should happen. Yes, Brooklyn. That is why certain traits in animals vanish. No, that has nothing to do with the dinosaurs. No, we're not talking about dinosaurs. I want to talk about dinosaurs. I love dinosaurs. We are not talking about dinosaurs. No, dodo birds died out because they got shot. Dinosaurs died out because they also got shot by something much larger. Like a friggin' asteroid. Earth got shot. Unrelated, wildly unrelated to what I'd like to talk about now. Okay, so that is a really brief introductory crash course with rabbit trails I wasn't expecting on where this genetics stuff can go. I said right at the beginning, I am going to grossly oversimplify things. That was my attempt at showing you where it could go. And it can go further than that. Ignore my daughter. No. We are not doing dihybrid crosses. I did start my little aside by saying, I just want to show you, I don't expect you to actually, I know Mr. Morrison's doing it with his pre-diplomas. Mr. Morrison's doing all sorts of crazy stuff with his pre-diplomas. Okay, find me this page. We're going to talk about codominance, incomplete dominance, and sex-linked traits. Let's do it. Let's talk about cows. Yes, moo moo. Okay, here we go. So, in homozygous dominant situations, nothing weird happens. It happens exactly the way you expect. In homozygous recessive situations, what you expect to happen, happens. Things get weird, though, in the heterozygous scenarios. So, let's talk about these. Codominance and incomplete dominance. These things tend to happen in heterozygous scenarios. Things start to get interesting. So, sometimes with some traits, in a heterozygous scenario, codominance can occur. Hands up if you've seen The Office. Okay? So you remember the short time, the storyline that they had where Michael and Jim were together branch managers. They were co-manager and co-manager, both fully managers, neither one totally the manager. What ship would set sail without two captains? Where would we be without the popes? Plural is the quote from that section. This is exactly the same. That word co, which is interesting now because we're actually picking a new pope for those of you who aren't Roman Catholic and don't know. That word co there means they both exist together in their fullness. Neither one gets to take over. So in codominance, both alleles, so both expressions of a particular gene, are fully expressed. What does this mean? How could this occur? So for those of you who are, is anyone in 4-H? Okay, so not Evelyn. You would know. It's like a club for people who like animals. Like raising, like husbandry for sheep and goats and cows and horses, and they do all sorts of stuff. So clearly no. Okay. I'll show you the coloring here because I wasn't able to print the other ones. This is a cow that is both white and brown. The name for that is what's called, sorry, clearly it's what? Yes, I do believe it was stretched. Not as much as you might think, but it was stretched. I don't know. I don't know a lot about the biology of cows. Cow part, like a gizzard from a bird, but not. I don't know. So this kind of coloring is what's called roan.
where you get the blending of the two colors, both showing up in their fullness. You have brown, you have white. There is no blending of the two. They're both there. That is an example of what's called codominance. Roan bulls or cows are heterozygous. Heterozygous. Not the first time we've written that word today. Where both the base color and white alleles are fully expressed. Brown being the base color, white being the color on top. One allele is expressed in white hair. One allele is expressed in brown. And codominant alleles are expressed, the way we write them is a little different than how we've been writing things up till now. When alleles are codominant, we express them using a capital letter for the gene with a superscript letter for the allele. Aha. That is literally what's coming next. So white hair and brown hair are both dominant. They show up together. And so the letter we're going to use for this is the capital letter for the trait. Let's call it hair. So a roan bull is going to be big H, big H. The dominance has been expressed. And we want to represent each color. Well, you've got... Ooh, I was wrong. Let me fix this thing. That's not supposed to be brown. That's supposed to be red. It's white and red, not white and brown. My bad. So you would write it as capital H for a dominant hair color with a capital letter R for what is the dominant color, red. And you have another dominant allele with a superscript or exponent like W. And you get a roan when you mate a red cow. So a fully red cow would be HRHR with a white bull, HWHW. And then the Punnett square would look like this. You've got HRHR, dominant red cow with dominant white male, bull, and you're going to get HWHR, HWHR, HWHR in every single one of those squares. So you're going to get 100% roan offspring. Whether or not they're male or female. And there's all sorts of, yes, Kaylee. The W comes from so the capital letter is the dominant trait. The R's and W's are the expression of that. So W for white, and it should have been red, not brown. I screwed this up right here. So the R for the redness and the W for the whiteness. No, it does not matter which one goes first. No, the H would be lowercase if there was a recessive trait there. Yeah. If there was a recessive trait that showed up in here, the H would be lowercase. Sorry? Would? Yeah, well, you wouldn't have a blended color. If you had a recessive allele pop in here, you'd have a full-on red cow because it would be a dominant red with a recessive secondary color. Okay, so an example of codominance is a disease called sickle cell anemia. This is an example of codominance in people. So this is a picture of what red blood cells are supposed to look like, these circular ones. They're supposed to be like circles but like indented in the middle. Increase some surface area. Sickle cell anemia creates some blood cells that don't form circles. They form these kind of crescent shapes. It is caused by a specific form of gene that directs the formation of something called hemoglobin. Hemoglobin. Hemoglobin's the next blank as well. Hemoglobin. Careful not to spell it hemoglobin. Hemoglobin is a protein in red blood cells that should be carries, not carried, carries oxygen. It's what causes oxygen to bind to red blood cells. Red blood cells that have that sickle shape do not transport oxygen effectively because they cannot pass through small blood vessels because of their odd shape. Unless they're oriented perfectly, they don't fit. And this leads to blockages in your blood vessels. It can lead to heart attack or stroke. And tissue damage. Tissue damage because blood vessels are getting torn apart, perhaps. Or tissue damage because there's parts of your body that need oxygen that aren't getting it. Hemo, H-E-M-O. Hemoglobin and hemoglobin, yes, H-E-M-O. So the allele for normal hemoglobin is represented as, we're going to add more letters, HB with a capital A up top here. That's what the normal allele looks like. And the allele for sickle cell is HBS. So you got hemoglobin A and hemoglobin S. Individuals who are homozygous I don't know. A for normal for some reason? I don't know. I just know that this is the notation that gets used. So individuals who are homozygous, like HBS, have full-blown sickle cell. Individuals who are heterozygous HB big A, HB big S, have some cells that are normal and some that have sickle cell. Yes. Sorry? Can you speak up? What happens to people with full sickle cell well, the easiest thing to say is that they don't have properly transporting oxygen in their blood. So you can connect the dots. If your blood isn't transporting oxygen the way it needs to be transported, the odds of you surviving are slimmer, especially if you're in the developing world. And interestingly enough, for reasons I do not know, sickle cell anemia seems to show up more in some populations than in others. Yeah, like racial groups. What do you mean, what is it? It's this. Yes, it is. Yes, it is very bad for you. They are red blood cells that don't form properly. They should all look like inner tubes. They don't. And so then these ones can't make their way through the smaller blood cells in your body. So they can't oxygenate you properly. It can lead to heart attack, can lead to stroke. So these people are said to have So these people, the heterozygous people, are said to have the sickle cell trait, but because it's codominant, they rarely experience any symptoms because they also have the dominant normal trait. So what this means is you're a carrier if you are heterozygous for this, but may not have symptoms. You may never have symptoms. I suppose it's theoretically possible that any one of you in this class could have a sickle cell heterozygous. You could have the gene, but you could be heterozygous with the regular gene, and you'd never know. Yes, Evelyn. No. This is more no. Well, this is genetic, and talking about physical structures in your body. Things like ADHD, autism are entirely different. But yes, I suppose you could go, but I mean, yes, you can. So the short answer is, yeah, you can go undiagnosed with all sorts of things as long as it's not impairing you. So we've got two people, two parents. She's got the sickle cell trait. He's got the sickle cell trait. But if we take a look at their children, let's say these are both, and look at this Punnett square here, they're both heterozygous. So let's label this here, HBA, HBS, HBA, HBS. You get all of the possible crosses. You've got 25% of your children could end up HBA, HBA. Totally normal blood cell production. You could have 50% of your children, this middle chunk here, could themselves be heterozygous, which means they have the trait, but they don't necessarily have any symptoms of the trait. And only 1 in 4, 25% of your offspring would actually have full-blown sickle cell anemia. So if you were heterozygous and you ended up with someone who was heterozygous, 3 of your 4, 75% chance your child is going to be totally fine. They might have the trait but no symptoms, or they might not have anything at all. But there's going to be then a 1 in 4 chance. And these are things you need to know as a parent. These are things that get tested for. If you're a parent who knows you're a carrier for a particular trait, and you know your partner either has the trait or has the full-blown disease or condition, it's not called a disease, it's called a condition, that's something to be aware of. Do you make the choice then to not have kids based on the likelihood that you're going to pass something on down? Certainly something to consider. Questions, comments, concerns? Genetic testing. Or, I mean, if you're talking about sickle cell, all they'd have to do is look at your blood, draw some blood, put it under a microscope, they could look at the red blood cells, and they'd know right away. But for the general, what about, how do I know if I'm a carrier even? Without doing a blood test, it would be a genetic test. And they wouldn't do the blood test on you or the genetic test on you unless they knew they had something to look for. So there could be millions of people around the world with the trait that are asymptomatic. It's like COVID. There was lots of people who got it, who were asymptomatic for it. So they infected other people, but they themselves didn't express symptoms. Happens with the flu, happens with the cold, but can also happen with these genetic traits. When you're ready, flip the page. So that's codominance. Both are fully expressed. Here is incomplete dominance. Also shows up in a heterozygous situation. Incomplete dominance is when neither allele can fully cover up the other, and neither is fully expressed. So if you're incompletely dominant, a heterozygote shows a trait, so someone who is heterozygous shows a trait that is somewhere in between the dominant and recessive traits. Someone who is heterozygous. So we, in incomplete dominance, we use the same method for writing the genotype as in codominance. So let's use the example of a flat, well, I'll show you, I'll fill in this here in a second. Let's use an example of something called a 4 o'clock flower. So just like Mendel's plants at the beginning, they're true breeding. So let's say we take a true breeding red flower and a true breeding white flower. If they're incompletely dominant, then we use the same writing as we did for codominance. We'd say that the color is red and the color is red. And they're both, it's a dominant trait. And for the white flower, we'd say the color would be white and the color would be white. So then if we bred these two flowers together, let's say one parent is CR, CR, and the other one is CW, CW. We get CW, CR, CW, CR, CW, CR, CW, CR. Do I have any artists in the room? Artists? Anyone taking an art class? Has ever taken an art class? Okay, Brooke. All the Brooklyns. Cool. What do you get when you take red and you add white? You get pink. So then each one of these, if it's a codominant situation, you'd get the blending of red plus white equals pink. But this itself can lead to all sorts of crazy things. We're going to do two more examples on the left and on the right. I hope you left some space. I'm going to draw two more Punnett squares. One there and one here. Let's take some other blended colors. According to color theory, if I take blue and yellow, what do I get? Green. So let's breed a green flower with a pink flower and see what kind of colors we can get. Green, if the colors are codominant, green would have to be what? Or CBCY. Right? Because blue and yellow make green. Pink, we've already established, is CWCR. Let's fill in the chart. This one would be CWCB. This one would be CWCY. This one would be CBCR. And this one would be CYCR. What colors have we just created? Top left, well, if you take blue and you add white, what do you get? A light blue, you betcha. You take yellow, you add white, what do you get? Light yellow. You take blue, you add red, what do you get? You get purple. Now all of a sudden we got some real cool phenotypes. You take yellow, you add red, what do you get? Orange. all you would then have to go back and do is figure out, well, what are the base colors? Like, what are the genes that are expressing these different colors? And you get some cool stuff. What if we took pink and white and bred those together? Or sorry, two pinks. Let's take the two pinks and breed them together. We're going to go CW, CR, CW, CR. What's going to pop out here? Let's just fill in the chart. What's going to pop out here and here? Those are both going to be pink again. So two pink plants would give you 50% pink offspring. And what about these other, what about the corners? Top left corner is going to be white. Bottom left is going to be red. So now you can start actually determining what the base colors are. If you breed a bunch of pinks together and get 50% pink and 25% red, 25% white, you know that white and red are going to have to be the reason the pink exists. So white and red then become the dominant alleles that are incompletely dominant in this case. And the last one, do I want to, I got 4 minutes. Do I want to go here? You know what? I don't think I do. I think you're right. I don't want to go. We will pause here. We've got complete, incomplete. We'll take a look at sex-linked traits tomorrow. You have no homework from me today. We're going to have some fun tomorrow, though, man. Oh, boy. Sorry? Bad C. Any of you who have not written the quiz for me need to come tomorrow during tutorial. When the bell goes, you may also go. Have yourselves a super duper day. If you haven't handed your dragons, get those in. Please. Get your dragons done and in. If you need to come to a tutorial for that tomorrow too, you're welcome to do that. Delete. Oh, whoops. Don't want to delete that. Oh, yeah, we do. Delete you and you. Okay. Yes. As in codominance? Because I hadn't started an example yet. This is what I was going to put there. It's the same idea. It's the whatever the two traits are with the capital letter dominance. This just shouldn't be there. Okay.